Could your DNA be performing calculations like a tiny computer? That is the fascinating idea behind molecular computation in cells. Inside every living cell, DNA and other molecules do more than store information, they also process it, making decisions based on chemical inputs. For instance, certain genes only turn on if specific molecules are present, just like a computer program that runs only when given the right inputs. This behavior is strikingly similar to logic gates in computers where conditions must be met before an action occurs. In this sense, your DNA might be solving basic logic problems all the time, deciding when to activate a gene, produce a protein, or respond to a signal. But how can molecules act like computers? Cells use a network of chemical interactions where proteins, RNA, and DNA work together in feedback loops, switches, and decision-making circuits. These molecular systems can perform functions similar to and/or and not gates, which are the building blocks of digital computation. Example, a gene might only be expressed if both molecule A and molecule B are present, just like an AND gate. This logic-like behavior means cells can filter signals, adapt to changing environments, and even remember past events. Analogy, it is like a biological circuit board where instead of wires and electricity, molecules pass messages through shape and chemistry, quietly solving problems to keep you alive. How can DNA and gene regulatory networks within living cells perform logic-like operations, processing inputs, making decisions, and producing specific outputs while operating in a chaotic, thermally noisy molecular environment? Cells contain elaborate networks of nucleic acids, proteins, and metabolites that interact through binding, catalysis, and conformational changes, resembling the logic gates and circuits of electronic computers. And synthetic biologists have even engineered genetic toggles, oscillators, and Boolean gates using transcription factors and RNA switches, demonstrating that molecular components can be wired to mimic digital logic. Yet, when one examines these systems more closely, deep paradoxes emerge about what it means for a cell to compute and whether such computation can ever approach the reliability and efficiency of silicon-based devices. So far, so good, until we probe the fundamental constraints that accompany molecular computation. Unlike electrons in a transistor, biomolecules diffuse randomly, collide unpredictably, and bind with affinities that vary over orders of magnitude. Any logic operation built on these interactions must contend with stochastic fluctuations. A single extra molecule of a transition factor can flip a genetic switch, introducing error rates far higher than in engineered electronics. Moreover, every binding and unbinding event is governed by thermodynamic and kinetic parameters, meaning that information processing in cells is inherently tied to energy dissipation and the necessity of waste heat production to erase molecular memory, as dictated by Landor's principle. At the most fundamental level, the analogy between cellular circuits and digital computers begins to fray. In electronics, logic states are represented by well-defined voltage levels. In cells, high and low gene expression levels are fuzzy distributions shaped by promoter strengths, ribosome availability, and nonlinear feedback. Attempts to design perfectly orthogonal genetic parts often run into crosstalk, where one regulator unintentionally influences multiple targets, undermining the modularity crucial for reliable computation. Even synthetic circuits that function in isolation can behave unpredictably when placed in the context of the full cellular milieu, revealing that the cell's native regulatory network exerts dominant control over any inserted logic. And then there is the matter of environmental influences. Temperature shifts, changes in pH, nutrient availability, and interactions with other cellular processes can all perturb logic operations. A synthetic toggle switch may flip at one temperature but become stuck at another. An oscillator engineered to produce regular pulses can dampen or enter chaotic regimes under stress, just as slight variations in applied stress determine the outcome of a hysteretic material. Minor fluctuations in cellular conditions can steer molecular computation down entirely different trajectories, making reproducible logic behavior across diverse cells and across time remarkably challenging. What then is really happening within cells that compute? The molecular computation paradox highlights the tension between viewing cells as programmable machines and recognizing them as evolved, robust, yet messy biochemical factories. While cellular networks can approximate logic functions, they do so through analog probabilistic processes that rely on redundancy, feedback loops, and population-level averaging rather than deterministic gate operations. The very features that make living systems adaptable, their ability to tolerate noise, exploit stochasticity for differentiation, and reconfigure networks in response to stimuli, also preclude the strict digital fidelity we associate with classical computation. Ultimately, exploring molecular computation in cells forces us to rethink the boundaries of computing itself. It challenges the notion that computation must be error-free and discrete, inviting a broader definition that embraces statistical decision-making, energy information trade-offs, and the exploitation of physical chemistry for processing. As researchers continue to engineer ever more complex genetic circuits, the paradox remains a guiding reminder. Cellular computation is not merely a miniature version of silicon logic, but a fundamentally different paradigm, one where life's ingenuity turns noise into a feature rather than a bug. The molecular computation paradox reveals that living cells can process information and execute logic operations using networks of biomolecules instead of electronic circuits. In these systems, DNA, RNA, proteins, and small metabolites function as logic gates, switches, and memory elements. Cellular computation occurs naturally through biochemical reaction networks that respond to inputs and produce outputs. This demonstrates that computation is not confined to silicon-based hardware and can emerge from the chemistry of life. In bacteriophage infection, a viral genetic circuit decides between lytic and lysogenic pathways based on molecular if-then interactions. It is like a medieval message relay where couriers pass signals and make routing decisions without any central computer. Electronic logic systems rely on rapid deterministic voltage changes in transistors to represent binary states. Biological computation, by contrast, uses variable concentrations of biomolecules and enzyme kinetics to encode and process logical information. These biochemical circuits operate more slowly, but in massively parallel and noisy environments where robustness is achieved through redundancy. This fundamental difference highlights how living systems harness stochastic chemistry for reliable decision-making. A genetic AND gate in bacteria produces a protein output only when both inducer molecules are present, mirroring an electronic AND gate. It is like two locks on a door. Both keys must be turned before entry protein expression is allowed. At the molecular level, specially designed DNA strands undergo strand displacement reactions that emulate logical operations. Complementary sequences bind or release in response to specific trigger strands, enabling computations such as addition or pattern recognition. These DNA circuits leverage predictable base pairing rules to implement Boolean functions without enzymes. This programmable chemistry shows how nucleic acids can serve as versatile computing substrates. In DNA-based calculators, hairpin structures open only when specific input strands are present, producing a fluorescent signal as output. It is like a set of interlocking puzzles where only the correct combination of pieces, DNA strands, fits together to reveal a picture. Output. Proteins often change shape upon binding small molecules, creating natural biochemical switches that control downstream pathways. These allosteric interactions form feedback loops and cascades, yielding ultrasensitive or bistable responses. Such dynamics allow cells to amplify weak signals, filter noise, and store memory of past events. These reaction networks thus function as molecular circuits that implement conditional logic. The lac operon in bacteria remains off until lactose binds its repressor, triggering gene expression only when glucose is depleted. It is like a thermostat that remains idle until the temperature crosses a set point, then flips the heating system on or off. Synthetic biology enables the construction of artificial gene networks that mimic electronic circuits inside living cells. By combining promoters, ribosome binding sites, and regulatory proteins, researchers tune circuit behavior to perform desired computations. These engineered circuits can act as oscillators, toggle switches, or multi-inwet logic gates. This approach proves that the principles of computation are universal across electronic hardware and biological wetware. A synthetic toggle switch in E. coli flips between two stable gene expression states in response to external chemical inducers. It is like wiring a simple electronic circuit on a breadboard to blink an LED when two buttons are pressed in sequence. Cellular logic often exhibits threshold effects where small variations in input concentration produce dramatic shifts in output behavior. Cooperative binding and feedback create nonlinear responses that can result in all or nothing decisions. These nonlinearities are crucial for processes such as cell differentiation, where a clear fate choice is needed. Thus, cells achieve robust decision-making through the same mechanisms that underlie nonlinear electronic circuits. In embryonic development, morphogen gradients trigger gene expressions cascades only once a critical concentration threshold is reached. It is like a crowd that remains quiet until the cheering reaches a certain volume, then erupts into a synchronized roar. Harnessing molecular computation opens the door to programmable cells that can diagnose disease, release therapeutics, or produce valuable compounds on command. Designer genetic circuits could detect cancer-specific markers and trigger apoptosis only in malignant cells. This transforms cells into living devices capable of autonomous, logic-driven action within complex biological environments. Such capabilities promise revolutionary advances in smart therapies and biosensing. Cartel therapies use engineered receptors that perform Boolean logic on antigen profiles, improving tumor specificity. It is like equipping a security guard with a checklist of conditions that must all be met before granting access. Killing a target. Accurately predicting cellular computation is difficult due to inherent cell-to-cell variability, environmental fluctuations, and crosstalk between pathways. Constructing reliable circuits requires detailed knowledge of kinetic parameters, resource competition, and evolutionary stability. Issues such as circuit leakiness, metabolic burden, and genetic mutations further complicate design. Overcoming these challenges demands iterative refinement and comprehensive modeling. Synthetic oscillators often lose regularity over time as host cells evolve to disable the metabolic burden of the circuit. It is like designing software for an ever-changing hardware platform. Updates and glitches constantly force rewrite. Future research aims to integrate molecular computation with multicellular systems, enabling tissues that coordinate logic functions across populations. Advances in cell-free systems, real-time monitoring, and AI-assisted design will accelerate the development of robust biological computers. These efforts could lead to living factories, smart therapeutics, and novel forms of bio-based information processing. Ultimately, they promise to blur the lines between computing machines and living organisms. Distributed genetic circuits in microbial consortia are being engineered to perform pattern formation and distributed computing tasks. It is like building a swarm of cooperative robots where each unit follows local rules, but together executes a complex global algorithm.